Sacred Heart College, Tirupattur. It's my privilege to welcome you all to this 5-day online faculty development program on global scenario and future prospects in life sciences, organized by the PG and Research Department of Biochemistry, Sacred Heart College. Welcome you, everyone. It's a great honor and pride in welcoming our resource person of today's FDP session, Dr. Maria Arokyanathan, Associate Professor, Head PG and Research Department of Biochemistry, St. Joseph College of Arts and Science, Autonomous Kadalur, Tissue Engineering and Regenerative Medicine. Hearty welcome you, sir. Thank you, sir. Thank you for your accepting our invitation, for your great support and being with us to share your knowledge, sir. Hearty welcome you, sir.
Thank you.
With immense pleasure, I would like to welcome heads of the department and faculty members of our institution and the other institution. Welcome you all. Now I request Dr. Shiva Ilango, sir, Assistant Professor, Department of Biochemistry, to introduce our resource person. Please, sir.
Yes, madam. Good evening to all. Now I am very happy and a great privilege to introduce resource person of the day, Dr. P. Maria Arokyanathan, Associate Professor and Head, PG and Research Department of Biochemistry, St. Joseph's College of Arts and Science, Autonomous Kadalur. Sir was completed MSc Biochemistry in St. Joseph College, Autonomous Tiruchirappalli, MPhil Biotechnology in Bharathasan University, and PhD Biochemistry Bioproducts in CLRI University of Madras. Sir was cleared CSR NET Life Sciences exam and also ASRB NET in Animal Biochemistry in 2001. Sir was specialized in biomaterials, nanobiotechnology, wound dressing materials, and drug delivery. Sir have 27 years teaching experience in St. Joseph's College of Arts and Science, Autonomous Kadalur from 1998 to till date. Sir was participated 54 seminars, conference, workshops, and presented 11 papers in seminar, conference, workshops, published in 5 book chapters, 21 papers published in journals, 44 PG students guided, 18 MPhil projects guided, 3 PhD scholars completed and 3 PhD scholars guided. Sir gave 15 invited talk and organized 14 seminar and workshops. Sir was reviewer in various international journals. Sir was article published in highest impact factor 10.7, site score 22.4. Sir was attended orientation program, refresher course, FDP, and completed bio nanotechnology NPTEL course also. Sir have 2 intramural projects amount rupees 25,000 were undertaken. Sir was controller of examination, HOD in biochemistry, member in finance committee, and chairman in BOS in St. Joseph College of Arts and Science, Autonomous Kadalur. Sir was subject expert UG BOS, PG BOS, University nominee in Sacred Heart College, Autonomous Tirupattur. Sir was subject expert BOS in EGS Pillai Arts and Science College, ADM College, Autonomous Nagapattinam. PG BOS member in Annamalai University. Thank you. Now hand over the session to Dr. P. Maria Arokyanathan, sir. Take over.
Thank you, sir.
Maria, sir, kindly unmute yourself.
Thank you, Maria.
Sir.
Maria,
Now you can hear me,
yeah.
sir.
Yes, sir. Yes, sir.
Okay. Good evening, everyone. It's really an honor for me to be the part of FDI program, to be part of all the faculty members who are joining here from various colleges. Really, it's a wonderful opportunity given to me by the Sacred Heart College management and the biochemistry department. So really, I thank them for giving this opportunity. Now I'm going to my sessions. My topic is, can you see the slides in full?
Yes, sir.
Okay.
Yes, sir. Yes.
So my topic is tissue engineering and regenerative medicine. This topic is really eye-opener to many one. and also is a very upcoming field. This tissue engineering and regenerative medicine, normally if you see, just a minute, the slide is, yes. So tissue engineering, it is not a single field. It is a interdisciplinary field which combines biology, engineering, and material science. So these three fields are combined together to develop materials, mainly the biological constructs, that is responsible for restoring, maintaining, and improving the tissue functions. So the main goal of this tissue engineering is to create functional tissues and organs by using cells, scaffolds, and growth factors. The coming to regenerative medicine is a very broad field that comprises tissue engineering also, but it's mainly focused on the repairing and replacing the damaged tissues by improving or inducing the own body healing mechanism. That is regenerative medicine. This involves stem cells, gene editing, and tissue engineering, and also biomaterials for regenerating the tissues. So mostly these two terms, they are interchangeable, but the broader area is regenerative medicine, which comprises tissue engineering. So now we are going to see these upcoming field, the tissue engineering, regenerative medicine, how it impacts our life, and what are the things they are doing nowadays, what are the avenues it is there, and where we can venture, and what progression is there, and what are the challenges we are yet to face, and future, really it will be a wonderful thing if all the challenges are met. So we will go into this topic detailing about this tissue engineering and regenerative medicine. So the tissue engineering is nothing but is an interdisciplinary field combining both biology, engineering, and material science. So this tissue engineering is a very rapidly growing field nowadays in biomedical science. This term tissue engineering was introduced in the year 1993 by Langer and Van Santi. So this is a field connected with, I told you already, the biological constructs or substitutes, mainly to maintain and improve and restore the tissue functions, and also to improve the tissue damage. Why we go for this tissue engineering? Okay, this is a field mainly for damaging or improving or restoring the tissues, we are using it. Why we go for this tissue engineering? Mainly because the first thing we go, why this engineering field or tissue engineering field have emerged, because of things, the lack of donor. Nowadays, we have so many patients, or in the globally, we cannot give them organ. Organ donation is a big problem there. Many die because of that. So the lack of donor for those who are having transplantation problems. Likewise, the efficient substitute for the organ. We don't have a efficient substitute. The organ is not available, we cannot get a proper substitute for that. That is another problem. And moreover, sometimes we can have mechanical devices or surgical reconstruction devices are available. And really it's a very boon. Those mechanical devices nowadays they're using in surgery and all these things. It's very, very important thing, because they save many lives using this. But in spite of this, they have a problem because they cannot accomplish all the functions of the natural tissues. Even though they cannot match the organs, natural organs, but they save the life, but they cannot perform functions that is the tissues, the functions performed by all the tissues. And also they're not very effective in controlling the progressive deterioration of the patient's cells. So because of this only, this field has emerged, tissue engineering. And really it is a very, very tremendous growth is occurring in this field. And really it is going to change current scenario. In few years, we can change a lot of changes we can see in this field, tissue engineering. So what is the importance of this tissue engineering? Mainly addressing the organ shortage. So the tissue engineering, I told you already, because organs is a big problem, because the global organ shortage is huge. We cannot meet that. So what is the alternative? The alternative is the tissue engineering. Likewise, the personalized medicine. So the tissue engineering helps to create customized tissues for individual needs and for the genetic makeup, we can make it for any type of allo rejections or any type of rejections, implant rejections or immune sensitized peoples. So these persons, we can make the engineering tissues from the own individual cells. We can grow it and that can be used. Likewise, improve treatment outcomes. So these tissues which we are engineering, they perform better compared to others, compared to other type of traditional treatments what is available. Thereby what happens? We can increase the quality of life. So this is the importance of the tissue engineering. So we can address the shortage of the organs and it can be used as a personalized medicine. And also the outcomes, the treatment outcomes is better compared to the traditional things what we are using it. So the main components of tissue engineering, what are the main components involved in tissue engineering? If you see, there are 4 things. The first is cell. This is the building blocks, whatever we make, the tissue engineering. But where these cells we can get, the cells can be derived from stem cells, it can be embryonic or adult stem cells, or the primary cells which we can derive from any type of tissues like skin, cartilage or bone. So these are the building blocks. So whatever, when we construct something, we need the primary material for constructing that. The primary material is the cell. That cell we can get it either from stem cells or from the primary cells obtained from the tissues. Then second one is scaffolds. Scaffolds is very, very important because they give the physical support to grow. They give the framework to grow. And they give the support to grow, divide and form the structures completely. So these scaffolds are either from synthetic origin or from natural origin. Natural origin, we have a lot of examples. Few examples I quoted here is collagen and alginate. These are the natural derived scaffolds. And the synthetic one, we have polylactic acids and polycaprolactone. There are a lot of things out there. They're all synthetic polymers. So these polymers, either it is naturally derived or synthetic derived, are mainly responsible for giving a support for the cells to grow and build up and so that the tissues can form. The next important thing in the component of tissue engineering is growth factor. They are responsible for the cell growth, differentiation and tissue formation because they are added to this only to enhance the development of the tissues and functional tissues. So mainly they are proteins because the proteins, other factors, they are responsible for cell growth and differentiation. And the last one, the component in tissue engineering is bioreactors. So whatever the cells, the scaffolds and growth factors, whatever we are adding, and they should be done in a controlled environment. That is very important. So the bioreactor is a place where we grow these cells in a controlled environment, providing optimal conditions, the temperature, pH, oxygen, everything, so that the engineered tissues grow properly outside the body. So these are the 4 important components of the tissue engineering, whatever we make, whatever things we do. So these 4 important components play a very vital role in developing the tissues or developing the constructs or substitutes, biological substitutes for this. Then the stem cells in the tissue engineering. I told the cells we have types. We have both stem cells and primary cells. The stem cells used in tissue engineering, there are 3 types that are used. One is pluripotent stem cells. I think everybody knows that because they have the ability to form or divide into any type of cell in the body. So that they pluripotent stem cells. The next one is induced pluripotent stem cells. This is something different. They are adult cells and they are reprogrammed to exhibit the pluripotency. That is very important here. So these are something like the somatic cells. We can say that they are made pluripotent by inducing or reprogramming using some factors. So they are offering a source of patient-specific stem cells. So this is the next stem cells. Another one is tissue-specific stem cells. So they are found in almost all the tissues, various tissues, and they can differentiate into specific cell type within the tissue, not any other tissue. So that tissues can be programmed and that tissues can be used for making the cells and the tissues constructs. So these are the 3 types of stem cells used in tissue engineering. And the biomaterial scaffolds for tissue engineering, there are 3 types of scaffolds used, biomaterial scaffolds are used. One is natural scaffolds, other one is synthetic scaffolds, other one is composite scaffolds. What is natural shuffles? They are derived from biological sources such as collagen, fibrin, and hyaluronic acid. I think it has been repeated. So they provide better, that is, biocompatibility. And synthetic shuffles, they're all engineered from materials like polymers, ceramics, and metals. The advantage here is they have good mechanical properties and degradation rates compared to natural shuffles. So the mechanical property of synthetic shuffles is more compared to natural shuffles, but the biocompatibility of natural shuffles is more because they are easily can be inducted into a body and can be degraded. But synthetic shuffles will have some issues with respect to degradation rates. The next important thing, the shuffles nowadays we are using is composite shuffles. Nowadays it's playing a very vital role because we are combining both natural and synthetic materials to achieve the optimal properties. That is both mechanical and tissue regeneration applications and degradation. All these properties, somewhat we are improving it. So these composite shuffles nowadays again they are making into another way by combining many natural materials, less synthetic materials, so that we can achieve the optimum level of degradability and mechanical support, everything. So these are the three important shuffles of biomaterial used in tissue engineering. So the bioreactors. So these are a small example for the bioreactor tissue cultivations. After the cell has been derived from the stem cells or the primary cells, so they are seeded into biomaterial shuffle because I have told you already the shuffle plays a very important role because they give the site for attachment and the framework for cells to grow, divide, and proliferate. So the next is the nutrition and oxygen supply. So the bioreactor provides a controlled environment for cell growth and tissue development because we should give a optimum level of conditions for the cells to grow. Otherwise, cells will not grow properly because you should match and mimic the natural environment. So the bioreactor is the one which can provide the nutrients, the oxygen supply, and all the necessary growth factors for the cells to grow properly. And so that it differentiates into a proper functional tissue. So these are the important components and the role each and every thing the components plays in tissue engineering. Coming to this applications of tissue engineering, we have many types of applications, starting from regenerative medicine, organ replacement, cancer research and drug development, cosmetic and reconstructive surgery, dentistry and maxillofacial surgery, musculoskeletal applications, wound healing, biomaterial development, research models, veterinary medicine, immunotherapy, biomanufacturing. We can see the application. See, it is a very vast applications of tissue engineering nowadays is taking place. And we thought maybe we come across many things, but we doesn't know these are all comes into play in tissue engineering. So we will see one by one these applications, how it works in each area. First, regenerative medicine. In this, we see the skin tissue. So there are many artificial skin grafts have been developed. That is for mainly for burn victims and also with the chronic wounds. They use engineered dermal and epidermal layers. So the dermal and epidermal layers, they will synthesize artificially and they placed on the wounds. And there are a lot of examples are available for this. One example I quote here is Apligraph that is in the market. So it is having a bilayer skin substitute. This is mainly composed of two things, keratinocytes and fibroblasts in the collagen matrix. So this is available. This is mostly used for venous leg ulcers. It is very painful ulcers. Even in diabetic foot ulcers, it is very painful. For these type of ulcers, they are using this. That is, after cleaning all these things, all the dead tissue, everything is removed. They can apply. So you can see the diagram on the left hand side. So this is Apligraph. So Apligraph, it is a collagen matrix. In this, we have keratinocytes and fibroblast. So they have this layer, it is placed on the wound site, and it is having a very good healing property, and it is approved by FDA. So this is one example how the skin tissue, artificial skin grafts have been developed with the natural materials. This is a part of tissue engineering. Then bone and cartilage repair. So the bone graft substitutes and cartilage regeneration for injuries, osteoporosis and degenerative disease with osteoarthritis. Even for repairing the bone, cartilage, tissue engineering plays a very important role. I have given the two examples for this, which has been the market now. It is infused bone graft. It is the company is Medtronic. So they contain a protein that is RHBMP2, that is recombinant human bone morphogenetic protein 2. So it is on the left hand side. The diagram is there, that is the graft. So these grafts, they stimulate osteogenesis. They are used in spinal fusion surgeries, also for treating bone defects in the skull and limbs. They're using this because this protein, which is present inside this graft, that is responsible for stimulating the bone formation. So the protein we are using this RHBMP2. It is in the market. And the next one is MACI. It is nothing but autologous chondrocytes, the cartilage cells. They utilize the cartilage cells for example, if you see the bone, and can see on the right hand side, the degradation or deterioration of the surface of the bone. You can see pores. So there are a lot of problems, the cartilage degradation or deterioration, and we can see a lot of pain in the many patients nowadays. So how we can regenerate this cartilage on the bone cells? So they use in the market MACI. This is having autologous cartilage cells. So they are cultured these cartilage cells on collagen membrane. Then what happened after surgically? Then you can see before and after. So this is the first diagram. Before you can see how porous it is, how the cartilage has been totally eroded. It is not there. Cartilage is not there. Now it has been filled with the MACI, that is the cartilage with collagen, so that it will repair, so that without any problem. This is a natural material. So this is another advancement taking place in case of tissue engineering, in case of bone and cartilage repair. You can see on some examples I have shown here. All are skin grafts. Different skin grafts of different materials. You can see on the right hand side, it is a hybrid of chitosan agarose skin substitutes. And we can have a lot of materials with the help of collagen, gelatins, we can help of cellulose. So many skin grafts have been developed nowadays. Each one is having some uniqueness. Each one is bettering. One is bettering the another. So every year or every month we can find new discoveries are happening in this wound healing category, and each thing is better than is coming out. So we can see in few years many products will be there in the market which can be helpful for the addressing these problems, that is the bone grafts or skin grafts, all these things, maybe competitive level. The next important thing in the tissue engineering is vascular tissue engineering. So they develop artificial blood vessels for bypass and cardiovascular surgery. One example I can quote here is in the market is Humacyte, HAV. This is the engineered blood vessels. They are made from human smooth muscle cells on the biocompatible scaffold. So see, this is a very advancement. They make tubes, blood vessels, using human smooth muscle cells, which is on the biocompatible scaffold. That is a framework where it grows. So these vessels they are using for these treatments, for hemodialysis and also peripheral arterial disease treatment. They're using this for vascular tissue engineering, surgeries, all these things. Next is nerve regeneration. Nowadays, they are also developing materials for nerve damage, spinal cord injuries. It is available in the market, but I think mostly in the Western countries they're using this. One is Neurogen. So Neurogen, it is made up of collagen tube. In this, we are having chondroitin sulfate. This mimic the Schwann cells of nerve cells, thereby it promote the development of axons. So regrowth and improve nerve functions. So this has been in the market, and we can. These are all not artificial things. All it is a part and part of our body, all the cells. So these materials are being used, and they are growing instead of going for artificial or synthetic one. Now they're utilizing our own cells and our own body cells for regrowing or reconstructing or repairing or those cells which are damaged. For all this, they are using it. I have just quoted one or two examples for each and every category, but there are a lot of examples available in the market nowadays. So this is one for nerve regeneration. Then corneal tissue. So for those patients who have vision impairment due to corneal damage or disease, they have engineered the corneal tissues. And this is done in Osaka University. So they have done these corneal tissues, engineered corneal tissues from the epithelial cells, corneal epithelial cells itself. They grow on the scaffold, then they transplant it to the patients, those who are having this problem, so that they restore the vision. So this is another advancement in the regenerative medicines. Then organ replacement. We have many bioengineered organs nowadays. They are developing artificial kidneys, liver, lungs. And all these things are developed using the patient's cells itself, not from other cells. Why? Because we can minimize the rejections. So all these artificial bioengineered organs can be developed using the patient's own cells. So the researchers have developed small miniature livers by culturing human liver cells on the scaffolds, and they have transplanted this liver into the mice, and they found that it is performing very well. And we can see two examples I have quoted. One is bladder. So urinary bladder, it has been constructed on the right hand side. The other one is the artificial kidney. It is partly mechanical, partly machinery is involved. But slowly they are developing into artificial kidneys in full swing, all in the trial stage. Next is heart valve replacement. Nowadays, we can see heart bypass surgery, then angioplasty surgery. We know that a lot of problem is there. But replacing the valve in the heart with any synthetic material, it's always a problem for. Even they will say it is having 10 years span or 15 years span like that. But these engineered biovalves, they are biocompatible, and also they prevent rejection and calcification process. So another important property of these engineered heart vessels is that they can integrate into our cells and minimize the need of future replacement. So that is big advantage. It's going to revolutionize the surgery protocols, everything near future. Then another very important thing is pancreatic islets for diabetes management. So nowadays, what they do, they engineered islet cells. They can capable of producing insulin. That is one thing in the market is ViaCyte Pick Direct. So what they have done is that they have taken the stem cells which can produce islets of parents. So alpha cells, beta cells, everything. So what happened? It is planted. So you can see in the right hand bottom we have one diagram. That is a device. So this device with stem cells, they are planted into diabetic patients. So the stem cells will form islet cells. They can capable of producing insulin, and they can cure even type 1 diabetic patients. So surely in very short period, the diabetic patients will have a revival, and no need to worry about that. So it will revolutionize the total field of medicines. Instead of taking drugs, they will just implant into our skin. That will be helpful for us in future. In cancer research and drug development. So these tissue engineering plays a very two important roles in this cancer research and drug development. One is the tumor models. So they will form a 3D engineered tissue to study how the cancer progress, how it metastasis, and what is the efficacy of the drug in the controlled environment. All these things we can study using this tissue engineering. Likewise, using our patient cells, we can create tissue models so that the treatment can be specific, it be personalized. So these two areas, very important area for tissue engineering. Then cosmetic and reconstructive surgery. So this bioengineered cartilage and soft tissues for facial deformities or injuries. I think we can know it a lot of these three areas: breast reconstruction, hair follicle reengineering. So it's a very costly process nowadays. We have known it. For example, those who have gone for breast cancer surgeries to make that after mastectomy to reconstruct the breast, that is the tissue scaffolds. They can prepare their own by the lab. They can prepare with the help of their tissues and they can be filled. Then likewise, the face that is below the lower part of our face, that is a part where most of the during injuries or accident, this part has been mostly damaged. To reconstruct this, lot of things is available in the tissue engineering process. They're using 3D technology, using bioengineered cartilage, soft tissues. All these things can be used for facial reconstructions, because in past we can say they will take the flesh and the muscles from some other parts of the body and they can do all type of facial surgeries. But because of this interdisciplinary area, tissue engineering, it will revolutionize the surgery protocol itself. Likewise, hair follicle engineering. We can construct the hair follicle, we can grow the hair follicle, we can grow the skin. And after growing the skin with hair follicle, we can patch that skin on the head so that it can grow. Because when the hair follicles die, the hair cannot grow. But we can make these hair follicles and we can grow the skin and hair follicles together, and we can patch this hair follicle along with the skin on the patients so that we can create new hair on the for example, nearly for treating alopecia. Alopecia is a condition where we can see there are certain areas where the hair will not grow. There are some type of alopecia is a condition. For example, we can say but normally hair grows everywhere on the head. But in alopecia condition, at certain patches, hair will not grow. There will be something baldness. This is called alopecia. So in that condition, this type of engineering can play a very vital role to reconstruct that. The next is dentistry and maxillofacial surgery. This plays a very vital role nowadays, because we know if you go to dentition, what are the problems? We cannot regenerate enamel, dentin, all this pulp, everything. It has been filled only. But now, this engineering, tissue engineering will revolutionize the total field of this dental. Because we can engineer the tissues, enamel, dentin, pulp, for the damaged teeth, and also for jawbone reconstructions. So the bone shuffles for the patients with the severe, for example, those who have gone for accident and some congenital defects, that can be reconstructed using this tissue engineering process. Likewise, periodontal regeneration. So some gum diseases, when gum has been a lot of infection, everything, the total tooth will come out. So all these problems of the dental things we have a solution in tissue engineering, and lot of work is going on in this area. And future, this dental field also will be revolutionary with tissue engineering process. Then musculoskeletal applications. The two important thing here is tendon ligament repair. Nowadays we are very prone. Previously we know only about bone fractures. But nowadays we know many will have this type of problems when we have some stress or stretch movements because of the ligament tear or the tendon tear like that. So for this, these tissue engineering shuffles tend to repair this tendons and ligaments. And a lot of clinical trials is going on. And very soon we will get some happy news regarding this, so that this will be the surgery and the other process will be somewhat non-invasive, will be occurring in near future. Likewise in muscle tissue also. So regeneration of the muscles for trauma recovery and degenerative conditions like muscular dystrophy. Muscular dystrophy, the muscles go on weakening. There is no other goal. So this type of patients, where this field will be a boon, because we will make them to muscles to grow, make them to grow, regenerate. That is another thing, is a big boon for them. Then wound healing. I think I have already mentioned in the regenerative medicines, a lot of films are available for wound healing. So like advanced dressing, it is there, like chronic wounds, like diabetic ulcers. So all these type of wounds, we have a lot of dressings that are available. We have thousands of dressings that are available. Each and new combinations is coming out each and every month. And some go on for trial, clinical trial. But now we can see that collagen sheets are available in all the medical shops now. The collagen we have seen only in the papers and research articles, but now we can see the collagen paper, collagen materials are available in the medical shops. It has come out. So slowly, many new wound dressing materials will come out with advantage, lot of advantages and healing power. I hope we will get it very fast in the market. Next is biomaterial development. Hydrogels and shuffles and biowings for 3D printing. So hydrogels and shuffles. Shuffle, we know that it is a framework where the cells can grow. So we are making a biomaterial. The biomaterial can be from a natural source where it forms a framework for the cells to grow. Because the material will mimic the extracellular matrix of the cell, and it promotes the growth of the tissues, everything. Hydrogel, it is something like a sort of jelly-like substance. We don't have a solid material like a gel or some mucus-like thing. So that gel will have the property. What is the advantage of hydrogel is something we can inject that also, hydrogel. That is another advantage of hydrogel, and it is always will be moist. So if it is moist, what happens? The scarring will be less. All these things is useful. Likewise, bioinks. So bioinks, the 3D bioprinting nowadays is a very emerging field. We know that 3D bioprinting, because we construct the organs. Each and every organ, layer by layer, with 3D printing, not 2D printing. 3D printing, we are printing the layer of cells. So for this, we use bioinks. What is bioinks? Nothing but living cells suspended in a gel or materials. So for printing, we use ink. Ink will print on the paper. Likewise, for printing the organs, what we do, we use the cells. For example, for cardiac muscles, we are printing, you put cardiac cells. The cardiac cells is loaded with some type of gels or other materials, and the art shape will be printed layer by layer. The cells will be printed on that, so that we get a total heart, a 3-dimensional way. So this biomaterial development, the bioinks plays a very important role for 3D bioprinting. The research models, these are one more advanced one, that is organ on a chip. So microfluidic devices nowadays plays a very important, because these microfluidic devices are the very special type of devices, something like a slide shape, only glass slide, the small glass slide. So these glass slide like device plays a very important, because we can study the organ function within that slide, because using a micro or picoliter of tissues or samples, studying the disease mechanism, the drug toxicity. It is a very interesting this chip, organ on a chip. It is a very advanced field, it's developing now. So this is a very important area in tissue engineering. Next level, it's moving on with organ on a chip. Then veterinary medicine. So animal tissue repair and conservation efforts. So what is the most important thing in this is that to prevent the animal, some tissue repair, we should make non-invasive type of things, because we should not torture the animals. What type of thing, for example, if you want to make a surgery and keep it inside something, it is very painful. Instead of that, if you inject a material into that animal, the material will go and heal. So these type of things it has been promoting now in tissue engineering. So they can inject that non-invasive type. So likewise, a lot of things is available in the tissue engineering, in the veterinary field also. And immunotherapy. If you see this, engineering tissues to study immune responses and develop therapies for autoimmune diseases and immunodeficiencies. So we know that we are developing the cells or tissues, everything, or constructs, everything. But whether, and apart from this, we also develop materials to study the responses, immune responses, and also therapies for autoimmunity and immunodeficiency, how to treat. So this is another area where tissue engineering plays a very important role. Then the final one is biomanufacturing. That is artificial meat and biosensors. Artificial meat. Now, instead of growing chicken, goat, everything, now the next level of food production is artificial meat production using tissue engineering techniques, so that we can grow the meat in the lab. And what is the advantage? It is very sterile, without any pathogen, and without all these things is available. So it has been approved in 2 countries, in Singapore and UK. So it has been in the market, and for the production, it has given the approval. So this is another next level of meat productions. Then another one is biosensor. The biosensor plays a very important role in medical diagnostic and environmental monitoring, because we have a biological system to detect any changes. That detection is transformed into any type of messages, maybe an optical one, or some signals, or any type of electrical currents, anything like that. So that sensor will tell you regarding the pollution or type of diagnostics, everything. So this also plays a very important role nowadays, the biosensors and the artificial meat. So what are the limitations? We have 7 limitations of tissue engineering. One is biological challenges, technical challenges, regulatory challenges, economical challenges, clinical challenges, logistical challenges, lack of functional maturity. What is biological challenges? The biological challenges, main thing we are having is immune rejection. For example, if you develop cells, allogenic cells, that is donor-derived cells or non-human cells, if you develop cells, and what is the problem? They may lead to rejection. That is another problem. We have the biological challenges is there. Another problem is vascularization. Sometimes skin, all these things, there is no problem. The blood vessels will grow. But there are some tissues, some organs, if you develop, the development of vascularization is very difficult things. So that is another challenge we are having in biological part. The next is that is tissue integration. The tissue integration is that it's a difficulty to integrate the engineered cells into the normal cells. That is another problem we are having, because of some other mismatching. All these things is there, but there is some problem in that. Then another problem we are having is that it's very difficult to develop complex structures like some organs, skin, and other things we can develop very easily. But when we want to develop organ, then that is a big problem. It's a really challenging. Now they are working on that. So these are important biological challenges we are facing. And then we can have technical challenges. In technical challenges, we have shuffle limitations. What is shuffle limitations? The main thing is that the shuffles, we have the difficulty in the shuffles in mimicking the mechanical and biochemical properties of the natural tissues. That is another problem. The technical challenges. Sometimes they don't mimic properly. the natural thing. Then another problem technical we are having is scaling up. Producing large amount when we need large amount of tissues for any type of injury or any type of action is very difficult. That is another problem, technical challenges. Another problem here is the standardization. We don't have a proper protocol in producing a type of tissue engineering products. Each one will produce something and the outcome will be different. So there will be a variability in the outcomes. So there is no proper standardization protocols for tissue fabrication. That is another technical challenges we are having. Then another problem with regulatory challenges. Regulatory challenges we have mainly three. One is approval and ethical. Other one is the classification challenges. So ethical issues. What is ethical issue? We know that when we use embryonic stem cells and animal testing, that is ethical issues involved. Regulatory challenges. Another problem in that the approval of these products takes very long time. That is another problem in regulatory challenges. And apart from that, the classification is another one important problem in regulatory. Whether it comes under biologicals, whether it comes under medical device, whether it comes under drugs. So this is another problem in the regulatory challenges. So that is another problem in the regulatory one. The next one is economical challenges. Economic challenge, we know that is high cost. High cost means lot of, we know that the big instruments, infrastructures and the growth factor, chemicals, what we're using for doing the tissue engineering is very costly. Okay, we can put high cost, but what about the market? We have limited market because this expensive product cannot be afforded by the lower income countries, or we can say middle income people. So only the top level people can be affording this, but other things. So that is another problem with economic challenges. And also the investment risk. So if you put, for example, upfront, initially you put a lot of investment on this, but what is the market? So without market, we cannot put invest on this. That is another problem in the economic challenges we're having for the tissue engineering products. Next is clinical challenges. The clinical challenges we have mainly three. One is long-term viability. Sometimes what happens, these shuffles or engineered tissues, the functionality will not be over for a longer period. So what we should do, we should inject some more thing. Where periodically we should inject. That is another thing. Then another important thing is that for this is the safety concerns. For example, if you use stem cells, sometimes what happens, it can form tumors. The risk of tumor formation will be there because of this proliferating effect. So that is another clinical challenges we are having. And also we are having clinical challenges is that mainly what? To make complex organs. I told you already. So skin, cartilage, all these things can be easily made. But when we go for a bigger one, that is a big organ development, with the higher complexity, then it's a really challenge in the clinical area. So this is the good clinical challenges we are having. And the next one is logistical challenges. So the storage and the transport, all these things is another important problem we are having. Then the last one is we are having is functional maturity. What is the functional maturity? Functional maturity, lack of functional maturity. It is immature tissues. So what happens sometimes, what happens when you develop the cells or tissues? They will not, they may lack full functionality. They not develop into full functional things. For example, I can put an example for you here is, you develop your muscle cells. But the muscle cells, when we incorporate, the muscle cells cannot do contraction. Likewise, the nerve cells, when you go for nerve cells, they cannot conduct signals. So the cells will be there. The cells will be formed. It can mimic or it can have the same type of all the things, but the functional problem will be there. That is one important issues we should address. Then another big important thing is that the time. Developing these tissues takes very long period. So that we should take into consideration. These are the challenges we are facing in tissue engineering. The future directions, mainly in the bioprinting technology, they are creating more complex tissues with the integrated vasculature. They're advancing now so that all the organs what they develop will be functional. Likewise, they develop immune tolerant scaffolds so that they improve the cell sourcing methods. Like there is induced pluripotent stem cells. So the induced pluripotent stem cells now they are making or developing into immunotolerant scaffolds. Likewise, nanotechnology microfluidics also plays a very important role in enhancing this vascularization process and also the native tissue environments. And another important problem here is cost reduction strategies. So now they are working on that because the too much of cost is involved means the market will not be too much bigger. The market is not bigger than what is the investment they put will not be come into play. So for reducing these strategies, they are working on automation and scalability, the manufacturing process. And also they are working some regulatory frameworks to have a proper standardization and collaboration, to also promote innovation on this tissue engineering fields. So the emerging trends we can see mainly in the three areas. One is the bioprinting, that is, which allows the creation of three-dimensional tissues. They have precise control over cell placement and scaffold architecture. So the bioprinting technology plays a very, very important role in tissue engineering. Next important thing is organ on a chip device because they mimic the function of human organs. So they are more accurate in testing the drugs and disease modeling. So this is another big breakthrough in tissue engineering. And the last one is AI. So they are being used to analyze large data sets. So to predict tissue behavior and also optimize the tissue engineering process. So these are the important emerging trends occurs in the tissue engineering. So I conclude with this. So this field, the tissue engineering, holds a very immense potential in medicine and also improving human health. So we can see as the field is advancing more and more, we can see more innovative treatments and therapies will be emerging from this. I hope millions of patients and people will benefit from this worldwide. So thank you very much for listening patiently. So if you have any queries, you can ask me. Thank you.
Thank you, sir. Thank you, Maria, sir, for your wonderful lecture and very, very informative and resourceful lecture on tissue engineering. Thank you for spending your valuable time, sir.
Okay.
Thank you
very much.
I just have
Thank you,
only
sir.
one
Thank you.
question, sir.
Thank you.
Sir, what is the level, or we can say, what is the state of India in this tissue engineering research? At what level we are staying?
And in India, we have some areas we have developed. For example, we have developed in collagen sheets.
Okay.
In collagen, we have developed a lot of work has been done and market. We have a lot of products. And also in mainly in cellulose materials, we are developing now. So
Okay.
this area we are developing. But the 3D printing in dentistry, they are working on that. And also in medicine, they are working, for example, the 3D printing on ribs and also the jawbone. So they are working because sometimes we cannot have each jaw will be different for persons. So we cannot have implants. So
Okay.
on studying that jaw structures, everything, they are printing the jawbones. And after printing, they are implanting it. So they are doing it in India.
Okay. Okay, sir. Thank you.
Thank you, sir.
Thank you, ma'am.
Sir, some of the questions from the participants, sir. Shall I read, sir?
Sure.
Sir, one of the participants said, artificial need good for children. It does not cause any negative effects.
No, actually, it doesn't have any type of disease things because it has been sterilely, we are culturing, we are developing that material. So only thing is that we have some nostalgic, or sometimes we can say we have some inhibitions, whether it is we can consume it or not. For example, like broiler chicken, initially when broiler chicken, somebody is hesitating to take that. Likewise, these meat, we don't have any bones, only meat with all type of features, all type of the things available in the meat is there. But we doesn't know what is the taste, everything. When it comes to the market, whether it is palatable to the tongue of the country. That is another important thing, because each country will have different type of taste buds. So that matters.
Yes, sir.
But regarding the safe, everything is totally, we are free from any type of infections, because it's all, everything is sterile.
Yes, sir.
Okay,
Thank
any
you,
other
sir.
question, ma'am?
Yes, sir. Yes, sir. Can bioengineered organ can causes any harm to humans, sir?
So it is, I think bioengineered organs, and now they are developing. All are in the clinical trials, and the main problem in bio organ is that vascularization. Because the skin graft, other things is very, very easy for them. But when you go for organ, the vascularization, because when you go, the thickness of the tissues is more, the vascularization is a problem. So they are working on that now, they're developing it. So it is all in the trials. It has not come into the market.
Yes, sir. One more question, sir.
Yes.
Artificial meats are considered oncogenic and shows antibiotic resistance. Is it biologically safe?
Can you repeat the question, ma'am?
Artificial meats are considered oncogenic and
No, it is
shows
not oncogenic. It is not oncogenic, because we are taking the cells, we are growing the cells with all necessary conditions, and we are developing the meat without free from any antibiotics or anything, because we are growing everything in a sterile conditions, in a lab conditions. So, and they packed in also in a sterile pack. So it is highly safe. But only thing is that whether we like the taste or not, it's yet to be decided.
Yes, sir. Thank you, sir. Last one question, sir. How the hormones on a chip can be effective in vivo, sir?
Organ on chip.
Yes, sir.
So organ on a chip is something, a microfluidic device. So that device is mainly used to test the organ functions in a miniature way. It is not something like implanting that. We are testing the organ functions in a miniature way, in a small slide itself. That is using micromoles to picomoles. Sample size is enough to detect the drug testing mechanisms, everything. So that is the advantage of organ on chip.
Yes, sir. Thank you, sir. Thank you very much, sir. Most of the participants reply. It's a very informative. It's a very knowledgeable session.
Okay, thank you very much.
Dear participants, the feedback link will be posted in the chat box. Thank you, Philip. Sir, yes, sir. Sir, yes, sir. Nia, sir.
Sheila ma'am, kindly go ahead. Go ahead. Kindly
Yes, sir.
go ahead.
Bioengineering,
Good.
can bioengineering organs can replace lungs and pancreas treatment?
Can
Questions.
I can't get you. Can you repeat, sir?
Can bioengineering organs can replace lungs or pancreas treatment?
Yeah, I think I have told you that pancreas. It is on the trial. If it is working out, then we can treat all the diabetic patients with a single surgery. So the islets will produce insulin. Even we can treat type 1 without drugs, because the cells will produce insulin directly based on the signaling. So it is very, very useful, and it has been tried in the Western countries, and slowly it will, I think they are developing with the help of some more things, with the help of CRISPR technology. Now they are working on that. So this pancreatic, I think I told you the diagram, the stem cells, which can produce the islets, which can be used for type 1 diabetes. Now they're working with CRISPR technology, so that all the diabetic patients can be cured with a single minimal surgery.
Okay, now thank you. Thanks for your nice presentation. Thanks a lot.
Thank you, sir. Thank you, sir. Thank you, Neha, sir. Feedback link will be posted in the chat. Kindly fill it. Will be activated few hours only. Thank you, participants. Participants are kindly requested to stay connected for tomorrow's session at the same time. Thank you all.
Maria sir, thank you sir. Thank you very
Maria,
much
sir, thank you, sir.
for your valuable and informative lecture on tissue engineering. Thank you for spending your valuable time, sir. Thank you, sir.
Okay, thank you very much. Thank you very much, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you.
Thank you all.
No, thank you, Nah. Thank you for your availability. Thank you.
Okay, thank you very much.
Thank you. Thank you. Sheila? Sheila, wind up பண்ணலாமா?
Yes, na, பண்ணிடலாம்.
Okay,
Thank you, na. Maria, sir, thank you, sir.
Okay, thank you, madam. Thank you, madam.
Thank you, sir. Thank you.
Thank you, sir. Thank you.
Thank you, sir.